For the first time, researchers have observed a mechanism that plants use to protect themselves from sun damage. For plants, sunlight can be a double-edged sword. They need it to drive photosynthesis, the process that allows them to store solar energy as sugar molecules, but too much sun can dehydrate and damage their leaves. A primary strategy that plants use to protect themselves from this kind of photodamage is to dissipate the extra light as heat. However, there has been much debate over the past several decades over how plants actually achieve this. During photosynthesis, light harvesting complexes play two seemingly contradictory roles. They absorb energy to drive water splitting and photosynthesis, but at the same time, when there is too much energy, they have to also be able to get rid of it. In a new study at MIT, researchers directly observed for the first time one of the possible mechanisms that have been proposed for how plants dissipate energy. The researchers used a highly sensitive type of spectroscopy to determine that excess energy is transferred from chlorophyll, the pigment that gives leaves their green color, to other pigments called carotenoids, which can then release the energy as heat. This is the first direct observation of chlorophyll to carotenoid energy transfer in the light harvesting complex of green plants. When sunlight strikes a plant, specialized proteins known as light harvesting complexes absorb light energy in the form of photons with the help of pigments such as chlorophyll. These photons drive the production of sugar molecules which store the energy for later use. Much previous research has shown that plants are able to quickly adapt to changes in sunlight intensity. In very sunny conditions, they convert only about 30% of the available sunlight into sugar, while the rest is released as heat. If this excess energy is allowed to remain in the plant cells, it creates harmful molecules called free radicals that can damage proteins and other important cellular molecules. The simplest hypothesis for how plants get rid of these extra photons is that once the light harvesting complex absorbs them, chlorophyll passes them to nearby molecules called carotenoids. Carotenoids, which include lycopene and beta carotene, are very good at getting rid of excess energy through rapid vibration. They are also skillful scavengers of free radicals, which helps to prevent damage to cells. A similar type of energy transfer has been observed in bacterial proteins that are related to chlorophyll, but until now it had not been seen in plants. One reason why it has been hard to observe this phenomenon is that it occurs on a very fast time scale, femtoseconds or quadrillionths of a second. Another obstacle is that the energy transfer spans a broad range of energy levels. Until recently, existing methods for observing this process could only measure a small swatch of the spectrum of visible light. In 2017, the research team developed a modification to a femtosecond spectroscopic technique that allows them to look at a broader range of energy levels, spanning red to blue light. This meant that they could monitor energy transfer between chlorophylls, which absorb red light, and carotenoids, which absorb blue and green light. In this study, the researchers used this technique to show that photons move from an excited state, which is spread over multiple chlorophyll molecules within a light harvesting complex, to nearby carotenoid molecules within the complex. By broadening the spectral bandwidth, the researchers could look at the connection between the blue and red ranges, allowing them to map out the changes in energy level. Once the carotenoids accept the excess energy, they release most of it as heat, preventing light-induced damage to the cells. The researchers performed their experiments in two different environments, one in which the proteins were in a detergent solution and one in which they were embedded in a special type of self-assembling membrane called a nanodisk. They found that the energy transfer occurred more rapidly in nanodisks, suggesting that environmental conditions affect the rate of energy dissipation. A better understanding of plants' natural photoprotection system could help scientists develop new ways to improve crop yields.